the revision video for higher level only for biology, and today I'm doing something slightly different because after recording the IB version of this video I decided that actually it wasn't as good as the A level version that I'd done. And whilst there is slight differences in what you need to understand in terms of the course, I actually think this the A-level video is a lot better because of the slight different more details. So the majority of this video is actually the A-level video, apart from the stuff at the end on something called Titan, and I think it'd be much more useful to use that. So if I refer to A-level in it, please don't worry about that. The key facts are all there ready for you to use. So this still is important, and you really do need to understand muscle contractions. I think the extra detail I've put in will help you do that. So the first thing you need to know is that muscle contractions are effectively important for all living things. Movement, is a function of life, and there are two distinct types of movement. So there's muscle movements inside the body, so that could be peristalsis, example swallowing food or pushing food through your digestive system, peristalsis, lung ventilation, so breathing in and out, heartbeat. And you see here some examples of that where we have the filter feeders here. The shark here might not be moving, but it might be moving water through its body and out of its gills. And then obviously the other side of that is locomotion, which is when the whole organism is moving. Organisms that can't move and stay in fixed positions are called sessile, and organisms that move around are called motile. So obviously number 2 would be linked to motile organisms only. Introduce to you, we are focusing on something called striated muscle. It's also known as skeletal muscle, but under a microscope it looks striped, and therefore it has this name of striated muscle. And those stripes are key because they're the different proteins that are going to be working together to allow the muscle to contract. Basically, we might call the cells, and I might refer to it as muscle cells or muscle fibers. So because the cells are very different to other cells, they're often referred to as fibers. They're surrounded by a cell surface membrane called a sarcolemma, so you should be using that word rather than just a membrane. And they contain many mitochondria and specialized tubes or tubules that store calcium ions, which we'll come on to later. And instead of being called endoplasmic reticulum, although you probably get the mark for that, we should technically call these the sarcoplasmic reticulum. And basically muscle fibers are in bundles, okay, surrounded by connective tissue, and we call these therefore we'll refer to as muscle fibers during this video. They have a good blood supply of oxygen and glucose, obviously, hopefully, to allow aerobic respiration via mitochondria. And you'll see the need for ATP throughout the muscle contraction stages in a few minutes. What do we mean by a sarcomere? So sarcomere is one individual unit of the muscle fibre or the muscle fibril, and it spans the distance between two Z lines. So let's have a look at this and try and explain it in a bit more detail. So one sarcomere is the distance between these two Z lines here, and the Z line is at the end of the fin filament. So you see these fin filaments in orange here where they end, that is the Z line. Okay, the fin filament is a muscle called actin, which we'll be talking about a lot in a few minutes. And then in the middle of the sarcomere, there is this red bit here. There is the thick filaments, which is the protein myosin, and that's one with the heads on it that we'll be talking about in a few minutes. So there's a couple of different bands that you need to know. So the Z line is the end of each sarcomere. You've got the I band, which is obviously here, but also over here, and that is just where there is fin filament here. Okay, and then you have the A band, which is the region where both the thick and fin filaments overlap. And then there is also the H zone, which is where there is just thick filament, which would obviously only happen when the muscle is relaxed and not contracted. Now, those are terms you need to know. You need to be able to remember this picture, this image. So I think we need to get a bit more. Again, just to give you some different versions of it. So if we start at the bottom here, I think is probably the best thing. Okay, so here we have let's have a look at here. So we've got the Z lines here, we've got the thick filament here, and the thin filament obviously over here. Okay, and remember we said the I band was where there was just thin filament. Okay, and this therefore is quite light on the if you were looking at it under a microscope. Where you get the A band, where there is crossing over, it's a lot darker. And when there's just, I suppose, the H zone, which is here and here, just myosin, then you get the A band. Okay, now why is this the case? Well, basically it's the case because of this. If you look at this, we've got A here. This is where actin and myosin are overlapping, so you get a darker colour. You get B here, which is the I band, which is just the thin filament, and then you get the A band, which is obviously got a bit of the darker colour of the myosin. So basically it's the amount of overlapping of the different proteins that causes the different colours in the sarcomere. Remember, the sarcomere is the distance between two Z lines in a muscle fibro. Old version of it, but I think quite a nice version of it again. So again, we've got the H zone. Excuse me, we've got the H zone, which is where you just have the thick filament. Okay, which gives you this sort of colour. You've got the A band, which is the actin and myosin overlapping. You've then got the I line, which is where you just have the thin filament, the actin, and you've got your Z lines at the end of the sarcomere. So again, A bands are dark when stained, as both actin and myosin are present. The I band is where you only have actin present, so it's lighter, and the H band is sort of in the middle because you only have myosin present. So that's it. Now, the key thing that we're going to be looking at today is, well, how does this sarcomere overlap? And effectively, when a muscle contracts, what we are trying to do is what the muscle is doing is moving the thin filament, okay, this way inwards, and moving this thin filament, the actin, this way inwards, okay, and therefore you're reducing the distance between two Z lines during contraction. So the myosin is thick filament and it has heads facing either side, which we'll talk about in the next few minutes. And then the actin is a globular protein, has two chains wrapped around each other, and we'll talk about that. So there's an image of the myosin, the thick filaments there with the heads, which are important. So I said to you a moment ago on the last slide, this is all about making this sarcomere decrease in size when we're talking about contractions. So the sarcomere contracts when the myosin heads form cross bridges with regular receptor sites on the actin, and they're spaced out. And then using ATP, don't think it's that simple though, we're going to talk about it in a few minutes, they move the actin towards the center of the sarcomere, making the actin and myosin overlap. So you can see here the distance between the Z lines is much greater when they are relaxed to when they are contracted. The distance between the Z lines becomes much less as a result. And the reason is not because of the thick filament moving. The thick filament sort of pulls along the thin filament, the actin, along. So you can see here the actin has moved from here and it moves inwards like that to the center of the sarcomere, reducing the distance of the sarcomere, making the two overlap more, and therefore giving it, I suppose, a bigger A band and a smaller H zone as a result. We look at how this actually works. The evidence for this is that the myofibrils appear darker when they overlap, and we can see this in contractions. As I just said a moment ago, the I band, which is the region where between the two Z lines becomes narrower, okay, because there is more overlapping between the two. The Z lines move closer together, shorten the sarcomere, and the H zone, where it's just in the middle, the thick filament, the myosin, also gets thinner as a result. So we can see this under a microscope during contractions of the muscle fibers. Structures are really key, and you need to know the difference between the two. You may see, if you look at older books, something an extra protein here called troponin, which I've actually blanked out here. You see it was on here. We don't need to know what troponin is for A-level biology. We just need to know about tropomyosin, which is an extra filament. So we've got two different main free proteins involved, I suppose. Myosin. Myosin is a let me just get my thing up here. So myosin is the thick filament. It has heads on the end, okay, and you can see how they're aligned in filaments here. There's lots of them together with lots of heads. And then you have actin, the thin filament. That actin has an extra protein wrapped around it called tropomyosin. So you need to know from the top of your head, you need to be able to memorize the fact that the fibrous protein myosin forms a thick filament. It has two protein types, a fibrous protein arranged in the filament with lots of tails, and a globular protein with two bulbous structures making two heads at the end. The thin filament is made up of actin, and actin is a globular protein made of long chains twisted around each other. So you can see the blue chains at the bottom there. And there is something called tropomyosin, which is a long thin thread wrapped around the actin that blocks its binding sites when we're not contracting the muscle. Let's have a look then at how this works. So if you need a break, it'd be a good time to have one now, because looking at muscle contractions is a long job. So let's start off with what happens at rest. So when the sarcomere is resting, when it is not contracting, the actin has binding sites that are blocked. As you can see here, if you notice these little dimples in the actin are the binding sites. They are blocked by a molecule called tropomyosin, and that prevents the attachment of the thick filament of the actin, of the myosin heads, to the actin receptor site. And that is essential for to be removed if we want to cause the contraction of the muscle filament. So let's go through then what actually happens during muscle contraction. The muscle has to be stimulated. Now this will be covered probably in another lesson by your teacher, but let's have a look at this. This is called a neuromuscular junction, and I'll go through it relatively quickly, but it's important you can talk about the stages of this. So neuromuscular junction will be at the end of a motor neuron, because the motor neuron connects to the effector. Remember, an effector which causes a response is always a muscle which contracts or a gland which releases a hormone. So the motor neuron, the action potential rises down the end of the motor neuron. The action potential causes then vesicles to fuse with the presynaptic membrane, and that causes the neurotransmitter, which is acetylcholine, still same as in a cholinergic synapse, to diffuse across the synapse. This side, however, we then have, instead of a postsynaptic, instead of another neuron, we now have the muscle fiber membrane, or the sarcolemma. So the sarcolemma has receptors on it, just like in a synapse in normal regular postsynaptic membrane, and that the acetylcholine will bind to those receptors. That then causes depolarization, just as before, so sodium channels open, sodium rushes in, and that causes a wave of depolarization to travel along the membrane. But as the muscles are adapted to have something called T systems, and you think, why are they called T systems? Well, if you just think about it logically, this looks like the letter T, okay? And the T systems, the impulse travels down the T systems, and that causes depolarization, which causes calcium ions to be released from sarcoplasmic reticulum, which is specialized endoplasmic reticulum. The calcium ions then bind to proteins in the muscles, and then that causes muscle contraction, which is what we're going to look at in a minute. So the whole muscle contraction is controlled by the availability of calcium 2+ ions. Now the next bit then is acetylcholine esterase. So just so you're aware of this, we're talking about it, that breaks down the acetylcholine that's gone into these receptors, and then that returns back to the presynaptic neuron using ATP. But obviously for this bit, we're not really interested in this section today. We're just interested in how this nerve contracts. the sliding filament mechanism step by step. And remember, if you get asked to describe the actions in a muscle contraction, this would be what you'd be expected to talk about. They would more likely ask you about neuromuscular junctions if they wanted you to talk about the previous slide. However, if this came up as an essay, you could link it all to do with calcium ions and the importance of them. So step one then. So this is the slide, the image I showed you before. So we have the muscle at rest, the sarcomere at rest. So at this point, the myosin filament, its head is not able to bind to the actin filament. And the reason is that the actin filament has tropomyosin blocking the binding site. So it is unable to attach to the actin receptor site. Now notice before we move on though, there is one thing I want to make sure you're aware of. There is ATP attached to the myosin head, and that, sorry, ADP I should say, and that ADP is important because without that the myosin head will not be able to attach to the actin. So let's move forward. So obviously from what we just talked about with the neuromuscular junction, calcium ions arrive, and the calcium ions cause the tropomyosin, which is the red bead-like string wrapped around the actin, to have a conformational change. So it changes the shape of the tropomyosin. Now that causes calcium ion, that is due to the calcium ions, and that causes the binding site on the actin molecule to then become exposed, which will allow then the myosin head to attach to the actin molecule. And then next stage, stage number 3, there is obviously binding. So the myosin head now attaches to the binding site of the actin, and we say this forms cross bridges. So make sure you're using that keyword. This is caused by the ADP molecule, which is attached to the myosin head. So the cross bridges are formed due to the ADP molecule on the myosin head. We then have something called the power stroke. So this is the bit where people get a bit confused. So imagine it's like a gun that is loaded and the trigger has been pulled back, okay, or the cock of the gun is supposed to have been pulled back, okay, and it wants to go off like an elastic band that's been stretched and it's ready to just be released. And when it's released, the elastic band gets smaller, and that is called the power stroke. So what happens is the myosin head then changes its angle and it moves the actin molecule along. So those thin filaments move towards the center of the sarcomere, making the Z lines move closer together. Okay, this releases the ADP as a result. The ADP has now been released. So now, because the ADP has been released, there is now room to attach a new molecule to the myosin head. So a new ATP molecule is now attached to the myosin head, and that detaches the head from the binding site, okay, and therefore the myosin head has now become removed from the actin binding site. We then hydrolyze the ATP, breaking it down into ADP and phosphate and inorganic phosphorus, okay, and that cocks the head or reloads the head, however you want to say it, of the myosin head. And here, if there is then more calcium ions, this will cause the ATP to be hydrolyzed, and that will allow that to happen to this higher energy position. The ATP has gone through hydrolysis, that has produced an ADP molecule and released phosphate. The ADP then does the same as it did before. It causes the head to reattach to the binding site. But whereas the head was here last time, the pointer is now, the ATP from the previous stage caused it to detach, and then it was hydrolyzed, recharging it to the cocked position. It's then left ADP, which will then which is then here. Okay, the release of the ADP will cause that to move along and push the actin filament this way a little bit more. Okay, and that will then cause the head to more ATP will cause the head to be released and then cocked again, and then that will go back here. So that constant switch between ATP being broken down to ADP, being released and then replaced by new ATP causes the movement of the actin molecule along this way in towards the center of the sarcomere. Notice the myosin isn't moving. Okay, so the thin filament, the actin moves, not the myosin. Now, all of this is being driven by calcium ions. So as long as the calcium ions are there, the contraction will happen. If they aren't there, the next stage won't happen. So why does that make sense? Well, when the nerve stimulation ceases, when the motor neuron prevents the acetyl, has no more action potential, the acetylcholine isn't released, okay, across the cytoplasm, the calcium ions get reabsorbed into the endoplasmic reticulum using energy from ATP hydrolysis. That means no more calcium ions are released. That prevents the ATP hydrolysis, and that prevents the head cocking and then reattaching due to the subsequent formation of new ADP. No more calcium ions also means that no more tropomyosin means tropomyosin doesn't go for a conformational change, and therefore it will go back and it will block the binding site of the actin again, preventing the actin and the myosin from attaching and forming cross bridges. And the force, and when the molecule, the opposite muscle, because remember they work in antagonistic pairs, contracts, that the force from that will pull the actin filament back out. Okay, so that's the detail you need to be aware. Just to give you some understanding of it, where does the energy come from this? Well, the energy comes from the hydrolysis of ATP, which moves the myosin head, as we talked about, and it also causes the reabsorption of calcium ions into the endoplasmic reticulum via active transport. Now, during intense exercise, you use aerobic respiration to make ATP and also anaerobic respiration to make ATP. But there is a third energy source that for A level we do not need to know a lot about, but it is called phosphocreatine. And basically you can replenish your ATP supply for a very short amount of time by splitting the phospho and the creatine up, so splitting the phosphate from the creatine to give that phosphate then to ADP to make ATP. Now, we don't need to know a lot about this at all. You just need to be aware that the phosphorus can be released from the phosphocreatine to make creatine, and that phosphorus can then join with ADP to make ATP. Now, phosphocreatine stores do not last very long at all, but we build them up in our bodies during rest, and then they'll be there ready for an emergency amount of energy if needed when we are working. Now, you might think, well, that's how it contracts, but how does the muscle relax? Well, muscle relax due to something called antagonistic muscle relaxation. And basically, when one muscles are working, they work around a joint, and the joint has an antagonistic muscle either side of it. So, for example, if I want to contract my bicep, my tricep is pulled. If I want to contract my tricep, my bicep is relaxed and pulled out. And the muscle that the protein that allows this to happen is called titin, which is the largest polypeptide discovered in humans. It is elastic and it effectively acts like a spring, releasing potential energy when it recoils. It connects to the end of the myosin and it has several functions. Firstly, it holds the myosin at correct positions in the center of the parallel actin filaments. It prevents overstretching of the sarcomere when the muscle is stretched, and it adds force of contraction by releasing energy when it Energy is needed to stretch that titin, and that happens when the muscle is relaxed to lengthen it. The energy cannot come from the muscle itself, but from the other muscle. So the antagonistic muscle contracts, which then pulls this muscle and stretches it out. And the contraction of one gives energy needed for the relaxing of the other. Video looking at muscle types and junctions, and this video is to look at particularly the different types of twitch fibers, but also look at neuromuscular junctions as a result. And this is updated for 2024 to fit the syllabus more accurately. There are lots of different areas that you'd need to look at if you're studying this in detail, but in terms of A-level, we study 3 different muscle types: cardiac muscle, which you study when you talk about the heart, skeletal or striated muscle, which is the muscle that contracts to allow you to move, and then smooth muscle, which is the muscle that pushes, say, the food through your intestines, for example. And this area, the syllabus focuses on striated muscle or skeletal muscle, which is the same thing. Makes up about 40% of the mass of your body. It has 2 types of fibers. We call them fast twitch fibers and slow twitch fibers. Basically, the difference is about how quickly they need to operate. So fast twitch fibers will operate rapidly, but for short periods of time, and slow twitch fibers can remain contracted for longer periods of time, but rarely have to move quickly. So a good example of a fast twitch fiber would be something like your bicep. If you're lifting weights, you have to move it quickly. Whereas something like your calf muscles would be a good example of a slow twitch fiber, which is something that keeps you stood upright all day. If you stood up in your job, you have to stand up a long period of time, but it's rarely that it has to move very quickly. Most people have on average about 50% of each. Some of the levels that you get can be inherited, but exercise and training will make you develop more of one over the other, depending on what you're training for. Numbers remain relatively constant, but the length of each fibre increases, as does the muscle cells, depending on how much you exercise for them. So you can see here we have the mean number of low fibers in leg or muscle arms. So for a cross-country runner, 79, whereas someone who's sprinting needs more fast muscle fibers, they have less slow fibers as a result. Okay. So if you're a marathon runner, you would have more slow twitch muscle fibers, uses lots of oxygen for fuel because it's going to work aerobically, and it provides continuous energy, offers extended muscle contraction, and basically fires slowly. Whereas if you're a sprinter, you'd have more fast twitch muscle fibers, which will rely on aerobic but also anaerobic respiration for fuel, and provides short bursts of speed, but it does fatigue more quickly. So let's have a look at slow twitch in more detail. There is only one kind of slow twitch fiber. They contain large oxidative enzymes, so lots of enzymes to do aerobic respiration. They're surrounded by more capillaries because they need a constant supply of oxygen and glucose to do aerobic respiration. They are highly resistant to fatigue, and they are more energy efficient because they're doing aerobic respiration, and they will therefore support endurance running. But if you train to be a sprinter, you can have 2 types of fast twitch fibers. They have small amounts of mitochondria. They're rich in glycolytic enzymes, so enzymes that do glycolysis, the anaerobic or first stage of respiration. They are fastest muscle fibers in humans. They have significant energy expenditure, so a lot of energy becomes wasted as heat, and they'll support quick. This, if we compare the two, so again, slow twitch is used in endurance, but fast twitch is used in bursts of activity. The contraction is slower, but for a longer period of time, which leads to low levels of fatigue, very slow fatigue in slow twitch fibers. So if you think about it, if you stood up, or your calves don't get tired for standing up. If you're stood up all day with one, often it's unlikely. So for example, for me, after the summer holidays, when I'm stood up in the classroom the first day, my calves might ache by the end of the day, and that's slow build up of fatigue. It's aerobic, so there's a high density of mitochondria and good blood supply for both glucose and oxygen. Don't just say oxygen. There's high levels of a pigment called myoglobin, which has a higher oxygen affinity than hemoglobin does, a very red colour, and this means it attracts oxygen into the muscle really easily, and it has low glycogen and phosphocreatine stores because it's aerobic, so it's constantly getting supplies of glucose and oxygen. Whereas a fast twitch fiber contracts rapidly and relaxes rapidly, but it fatigues quickly, so you can only use them for short amounts of time. They'll often respire anaerobically and therefore don't need the mitochondria, because remember, glycolysis, the only stage in anaerobic respiration, happens in the cytoplasm of the cell. Blood vessels are needed less because little oxygen is needed because it relies on anaerobic, and then it can slowly replace the oxygen to break down the oxygen debt afterwards. And it has very little myoglobin because little oxygen is needed, so they're often paler in colour, but it will have a lot of glycogen and phosphocreatine stores. And if you don't know what the phosphocreatine stores is, watch my video about muscle contraction. Compare the two here, you can see there are lots of comparisons, but let's just focus again on slow muscle fibers first. So slow muscle fibers are long, have a long contraction and relaxation style. They have a smaller store of calcium ions because they're working for a longer time, and therefore they don't need to constantly replace them and reuse them and release them and reuse them. They have lots of mitochondria and gain energy from aerobic respiration, as it has enough time to make ATP. They have a dense capillary network and mitochondria near the surface to reduce diffusion distance, and they contract slower, so need less amounts of calcium. They do suffer less from fatigue, as aerobic respiration means less lactic acid is formed, and therefore they suffer. To that then, the fast muscle fibers have slow walk for a short period of time but with more power. They need fewer capillaries because they rely on anaerobic respiration, glycolysis and phosphocreatine, so need less oxygen, and as I said, that slow supply of oxygen between exercise is enough to break down the lactic acid over time. They need high amounts of calcium ions because they need to initiate more contractions, so sudden rapid contractions needing high concentrations of calcium ions. They have high levels of glycogen and high levels of phosphocreatine stores as a supply of fuel for high intensity contractions, so when they need that fuel they need it quickly. And they tend to get fatigued more because they're anaerobic to generate lactic acid, which does reduce the pH. So to summarise, slow twitch fibres contract slower and provide less powerful contractions, but over a longer period of time. It's common in muscles like the calf muscle, which contracts constantly to maintain the upright position. Aerobic, and therefore it prevents the buildup of lactic acid. It's got a myoglobin store as an adaptation, which is good at storing and has a high affinity for oxygen. It's got rich blood vessels to supply lots of oxygen and glucose for aerobic respiration, that should say, and it's numerous in mitochondria providing ATP from aerobic respiration. Twitch muscle fibers contract rapidly with a more powerful contraction but over a shorter period of time, and it's common in muscles that have to have bursts of activity such as biceps, lifting weights, or for sprinters in their legs maybe. They're adapted by having thicker and more numerous myosin filaments, which means more myosin heads to attach to the receptor sites and therefore a strong contraction. They have high glycogen concentrations as energy store. They have anaerobic enzymes, so allow anaerobic respiration, and they have phosphocreatine stores, so the phosphocreatine can break down to phosphorus and creatine, and that inorganic phosphate can attach to ADP to replenish ATP oxygen. If you look at different people with different activities, endurance runners, so for example marathon runners, they will have a higher number of slow twitch fibers, which means they can continue their intensity for a longer period of time. They have a higher capillary numbers to allow that, which means they have higher oxygen levels and oxygen consumption because they're doing aerobic respiration. Whereas if you're doing something like sprinting, you have less slow fibers and more fast fibers, which is intense, but you need less oxygen because you're relying on anaerobic respiration and phosphocreatine to replenish that ATP. So just some little bits here, but you need to know that muscles obviously all work around joints. We attach muscle to bone via tendons, which is flexible but not elastic, so they pull on the bone. And muscles work by being antagonistic. Now what this means is when one muscle contracts, the other muscle relaxes. So the bicep, if this contracts, it stretches out the tricep. If the tricep contracts, it stretches the bicep, and that allows the movement either side of the. This is really probably extra information if you're doing A-levels more IB biology, but just so you're aware, you might wonder, well, why doesn't the one muscle, when the one muscle's been stretched, why doesn't it just keep stretching out? And that is because there is actually an extra filament called titin, okay, which is a muscle filament, and basically it stretches when the muscle is being stretched, and that muscle is, the energy for that is being pulled from the muscle that is contracted on the other side of the joint, and the titin basically only allows it to be stretched to a certain length. You need to know about neuromuscular junctions and how action potentials cause. You should be able to talk your way through this. Press pause and use the bullet points if you need to. But an action potential arrives at the presynaptic neuron at the end of the motor neuron. Okay, and remember the muscle is an effector. That action potential arrives, causes calcium ions to fuse in, which causes the vesicles containing the acetylcholine to do excitosis and release the acetylcholine into the synapse. That acetylcholine then diffuses across the synaptic cleft, and there are receptors just like in normal synapses on the membrane of the muscle. The membrane of the muscle cell is called the sarcolemma, and it has sodium channels which open when the acetylcholine attaches to it. That causes depolarization, so sodium ions rush in to the muscle cells, and then a wave of depolarization travels down the membrane until it gets to something called a T system, which is an invagination effectively of the sarcolemma. That causes then a depolarization of something called the sarcoplasmic reticulum, which releases calcium ions, which causes the muscle to contract. And that will continue for as long as the acetylcholine is being released. When the muscle contraction stops, when the impulses stop, those calcium ions will be actively reabsorbed into the sarcoplasmic reticulum using energy. Now, to stop the muscle contraction, the acetylcholine, which is in these receptor cells here, must be broken down and recycled. So an enzyme called acetylcholine esterase choline, which allows the sodium channels to close. So the acetylcholine is recycled using acetylcholine esterase, which breaks it down into ethanoic acid and into choline. These then diffuse back into the presynaptic membrane, into vesicles, using ATP to do so, and then that can be released when the next muscle contraction is needed. You might get asked to compare synapses with neuromuscular junctions. So here is this, but you might need to look at this in a little bit more detail in your book. So if we look at a synapse, it uses acetylcholine as a transmitter. So does a neuromuscular junction. Obviously, cholinergic synapse is used between two neurons, so between the sensory neuron, the relay neuron, and the relay neuron and the motor neuron, whereas this is always between the motor neuron at the end of the reflex arc and the effector, which is the muscle. Synapses can be both excitatory or inhibitory, whereas neuromuscular junctions can only be excitatory because cause muscle contractions. And whereas a cholinergic synapse is stimulated by an axon potential, so is a neuromuscular junction, and that shows you the link of the two and the system and how it works. So if you've got a question about the importance of synapses, this would be two of those.